So we are pleased to attend for the defense, PhD defense of Lorenza Pacini, and she will present her PhD work very soon. Before that, I'd like to introduce to you all the members of the jury. So it's a real pleasure first to have a defense in real, so that's a very good thing, and I'm very happy that we are back to normal, almost. And so let's start. The member of committee is composed of Ozlem Keskin, who is in video conferences. So she was also the reviewer of theses. David Leitner, who was also the other reviewer of the thesis, who is professor at the University of Nevada. Luisa Di Paola, who is professor at University of Rome. Maggy Hologne, who is a maître de conférence at the Université de Lyon. Christophe Menezo, professeur des universités at the Université de Savoie Mont Blanc. Claire Lesieur, who is chargée de recherche at CNRS and who is the PhD, one of the two PhD advisors of Lorenza. Laurent Vuillon, professeur des universités à l'Université de Savoie-Mont-Blanc, who is also a co-advisor of Lorenza. And myself, Nicolas Schabanel, who is directeur de recherche at CNRS. So you will have 45 minutes, about 45 minutes to present your work, and then there will be some questions.
So thank you very much. I will now present the summary of the results of my PhD project entitled Investigation of Protein Structure Sustainability and Bioinspiration for Urban Systems. This project was supervised by Claire Lesieur and co-supervised by Laurent. The goal of this project was the exploration of the design of sustainable spatial systems. Sustainable systems are those that remain functional in time. That means that they have to cope with perturbations that can be internal, changes in the system, or external, coming from the environment. In particular, I have focused my attention on spatial systems that are the ones whose function depend on how the components are arranged in space. I have studied two types of spatial systems, obviously extremely different one from another, that are proteins and cities. On one hand, proteins are sustainable because they have been functioning on Earth for a billion years. That means also in very different environments. So they are a good model to investigate how the design of a structure leads to sustainability in the system. And you can see an example of a protein structure here on the left. On the other hand, cities may be sustainable or not. Intuitively, we would say that they are not sustainable as we see pollution accumulating in the city, get traffic. And because cities are so different from proteins, they are also a good challenge to the possibility for using similar design criteria to design sustainability in very different spatial systems. So, what are the requirements for a system to be sustainable? If it has to cope with internal perturbations, it means that the system somehow has to be robust to these changes, and this can happen through the existence of alternative functional solutions, but also through the implementation of error correction mechanisms that can restore a functionality after it is lost. Moreover, sustainable systems also have to remain functional even when the environment changes, and this means that somehow they have to be evolvable to adapt to those changes that come from the environment. This can happen through internal perturbations that can modulate the function of the system and thus promote functional innovation. In my project, I have focused on only one type of perturbations that are internal, and specifically, I have studied the possibility for performing substitutions of components. In proteins, the components are amino acids, and you can see some examples of them in the protein structure. So a perturbation in a protein will be an amino acid mutation, that is the substitution of one amino acid with an amino acid of a different geometry. I have modeled cities simply as a spatial arrangement of buildings in space. So a perturbation will be a building reconstruction, that is the substitution of one building with the building of a different geometry. So I show here two examples of substitutions made in proteins on the left and in cities on the right, where the red component is substituted with a component of a different geometry. Just looking at the figures, we understand that the possibility for performing this substitution depends on how all the other components in the system are organized in space, and specifically whether they leave enough empty space in the neighborhood of the component to be substituted. And this allows performing substitutions, but also determines what are the possible dynamics in the system before and after the substitution. So we understand that we need a measure of space occupancy in the system that takes into consideration the proximity between all the components. So I will show some methodology to do so in these two systems. However, I will first spend a couple of words more on proteins to understand why they are sustainable. So as I said, proteins are made of amino acids. These are molecules that exist in different types, and so they are made of atoms. When the proteins are synthesized in the organism, they are made as a chain of amino acids, whose type and order defines the so-called amino acid sequence of the protein. And you can see an example of sequence on the top of this slide, where each letter corresponds to an amino acid type. The amino acid sequence encodes for what will be the final three-dimensional shape that this amino acid chain will take in space, and this is defined as the protein structure. You can see an example of protein structure on the left of this figure, where amino acids are colored based on their type, and spheres represents the atom of the amino acid. The protein structure, in turn, will encode for the possible atomic motions in the system, and we call this motion the protein dynamics. And then these dynamics will provide a specific biological function to the protein. So we see that the function of the system is encoded by the way the components, here the amino acids, are arranged in space by the structure. So in these schematics, I represent in a very simplified way the relations that I showed before. We start from the amino acid sequence. This encodes for structure that control the dynamics of the system, and this provides a biological function. Now, the question is what happens when substitutions of amino acids take place. In this case, we speak about protein sequence variant, because the sequence has been changed because of the substitution. And this the new sequence will encode some structure and some dynamics. And if this provides the same biological function to the protein, then we will say that the protein is functionally robust to the substitutions. And in particular, and we can also say that sequence 1 and sequence 2 are alternative functional solutions. On the contrary, other substitutions can lead to a functional change, and as we saw, this is important for the evolvability of the system. Finally, other substitutions can restore the original function of the protein. So in this case, also sequence 4 will be an alternative solution for that given function, and we recognize an error correction mechanism. Now, all these features are known to exist in proteins, and these are the keys to the sustainability of these systems. The question is, how does the design of the protein structure give such sustainability features to the system? And all the part of my project around protein structure sustainability was developed starting from this very generic question. So why studying protein structure sustainability? This is relevant, of course, in the field of biomedicine, and in particular for the development of personalized medicine. Indeed, personalized medicine has the goal of providing personalized drugs and diagnostics to individuals based on the exact sequences of the proteins that are produced by their organisms. And these sequences are different because of the different DNAs across different individuals. So if we understand how an amino acid substitution impacts the way the proteins function according to different sequences, then we may understand whether two individuals can be cured using the same drug or not, and if they have the same risk to develop some diseases. Another field where protein structure sustainability can be applied, it's a completely different field, and it's the one of bioinspiration. This is a design technique that consists in using a natural system with a specific property as a model to design man-made systems that would have the same property. In particular, if you understand how the protein structure design leads to sustainability, then one day we may use this information to design man-made sustainable spatial systems, as for example, urban structures. While studying urban structural sustainability, this is important nowadays because cities are facing more and more challenges, so there is in general a need for designing more resilient and adaptable urban systems. Some of these challenges come from urban population growth, that among other issues implies that we will have to house more and more newcomers in the cities. So this can be done in mainly two ways, either adding buildings outside the city, but this consumes land and this is not sustainable. And the other possibility is to replace existing buildings with buildings of bigger sizes. So in my project, I focus on this last very specific question, that is, can we substitute buildings in the urban systems as one aspect of the general urban sustainability problem. So based on these motivations, I have restricted my research to mainly three questions. The first one is, when a substitution is needed, can the new component simply substitute the old one, or is there a need for structural arrangement? And if yes, is this local or global? The second question is whether this arrangement impacts the dynamics of the system. And the third question is what happens when many substitutions are performed? What is their collective impact? As you can see, I do not mention the functional impact of substitutions in these questions. The reason is that I have preferred to look at the impact on the structure and on the dynamics before focusing on a diagnostic on the functional impact of substitutions. So I will take back now the picture that we saw before, where we had recognized the role of the spatial proximity between all the components as an indication on whether a substitution can be performed or not, and on its dynamical impact. So the fact that the relations between all the components of the system are essential to explain a global outcome, that is the impact of a substitution, this is a characteristic of the so-called complex systems. These are defined in general as those systems whose global behavior cannot be explained as a sum of the individual behaviors of the components taken as isolated. Because of this reason, I have used a modeling approach, which is borrowed from complex system science, in order to study protein and urban structures. This approach uses complex networks as a model of complex systems, where a network is what mathematically is represented by a graph that is an object composed of nodes and links. The nodes represent the components of the system, and links represent relations between them. Moreover, the links can be weighted according to some definition of importance of these relations. In my case, I will speak about spatial complex networks because the nodes have some coordinates in space, and the links represent spatial relations. Moreover, I will use link weights to quantify the quantity of space occupied by components involved in the links. So we will use spatial complex networks to measure space occupancy in the systems, and the goal will be to be able to distinguish neighborhoods, as the one that you can see in the schematics, where the number of components may be the same, but space is occupied differently because of the different distance between components, for example, case 1 versus case 2, the different orientation, case 2 versus case 3, or different geometries, as cases 4, 5, and 6. So to do so, I have used the amino acid network model to study protein structures. This is a well-known model in structural biology, and it is made starting from the crystalline structure of a protein that provides the coordinates of the atoms of the amino acids of the protein. So in this amino acid network, the nodes are the amino acids, and the links exist between amino acids that are at distance lower or equal than 5 angstroms, that is a threshold for chemical interactions. Moreover, the links are weighted based on the number of these atomic interactions between the amino acids. So in this way, the link weight will depend on the distance between the amino acids, their orientation, and also their geometry. For what concerns cities, I have proposed a novel model that I called the building network, that is constructed from the building footprint of an urban area that is available on OpenStreetMap. I will call the nodes of the building network buildings. This is not exactly true. They are rather convex halls of adjacent buildings. However, what is important is that they represent a geometrical entity that occupies space. So links exist in the building network between two buildings that are at distance lower or equal than 30 meters, and they are weighted according to the procedure schematized in the bottom of this slide. In particular, if we have two buildings BI and BJ that are connected, then we look in the 30-meter surrounding around each of these two buildings, what is the part of the other building that is touched. This gives the two red areas in the figure, and the link weight will be given by the sum of these two areas. So thanks to the definition of the link weight in the building network, we have again that the link weight will depend on the distance between the buildings, their orientation, and also their geometry. So using these definitions of link weights, we can discriminate the neighborhoods that we saw before, where space is occupied differently. And so in particular, we will use node measures in the spatial networks to characterize the neighborhood of some component I that is surrounded by some components J1, J2, J3, etc. So these are the neighbors in the spatial network of I. So in particular, we define the node degree, K of I, the number of neighbors of I. But as we saw, this does not take into consideration how space is occupied by these components. So this is instead provided by the node weight measure, W of I, that is defined as the sum of the weights of the links that I performs with its neighbors. So because of the definition of link weights, we'll have that a higher node weight corresponds to a more packed neighborhood, more space is occupied, while a low value of node weight for the same I will correspond to a loose neighborhood where some empty space is present. The problem is that the node weight measure does not allow comparing neighborhoods of components that have different sizes of geometry. For example, if we have a bigger component, it will have a higher potential for performing high weight links, but also a higher potential for having many neighbors. This is a high node degree. So because of this reason, we use a third measure called the neighborhood watch, and W of I, that is defined as the node degree, the node weight, normalized by the node degree. So mathematically, it is simply the average weight of the link that I performs with its neighbors. And it allows comparing packing of neighborhoods around components that also have different sizes or shapes. So in particular, we have a higher neighborhood watch value if the neighborhood is compact around I, while a loose neighborhood will result in a low value of neighborhood watch. So we can pass to the results, starting with the first question that was, can we simply replace the component, or is there a need for structural arrangement, and at what scales? I have defined local spatial sustainability, the possibility for accommodating substitutions without structural arrangement. And I report here some examples of neighborhoods of components that go from a loose case on the top down to a very packed case in the bottom. And as you can see, the possibility for substituting the component depends on how the neighborhood is packed. In the loose neighborhood, the blue one on the top, there is enough empty space to accommodate the substitution. This is still true in the case in the middle. Instead, in the case in the bottom, there is not enough empty space to accommodate the substitution. Some structural arrangement will be needed. So according to our definition, we say that the first two cases are sustainable and the last one is not. We have just seen that the neighborhood watch measure gives an indication of how packed is the neighborhood around the component, and so we have used it as a diagnostic measure for this local spatial sustainability in the systems. You can see here some examples of building networks built for 3 cases of urban structures: Mont in Lyon, Schabanel also in Lyon, and Manhattan in New York. On the top, you can see the building network models, and in the bottom, I have colored the buildings based on the value of the neighborhood watch. So we go from blue, that is the lowest value, up to red, that is associated to highest values of neighborhood watch. And you can see that there exist very different situations in terms of building layouts, giving both sustainable and unsustainable solutions. For example, in Mont, most of the buildings are blue or green. That corresponds to sustainable solutions because the parking is relatively low. So according to our definition, they are locally sustainable as they can accommodate substitution. On the contrary, in Schabanel, but even more in Manhattan, we find more and more red buildings that are associated to unsustainable cases at the local scale because the neighborhood is too packed to accommodate substitutions. On the contrary, in a database of protein structures, and I put some examples here, we have found that the values of neighborhood watch are usually moderate. That means that the packing around amino acids are never too loose, but neither too packed. And you can visually see this here from the fact that almost all amino acids are green. There exist, of course, some exceptions, but they are rare and they are well scattered in the system. So according to our definition of local sustainability, we say that proteins are sustainable. And so we expect amino acid substitutions to be possible in protein structures. Now, in some cases, the local neighborhood may be too packed to accommodate the substitution, but this may be possible through some structural arrangement. And we define this possibility the multi-scale spatial sustainability of the system. In the figure here, we go from a case that is locally sustainable on the top, where there is no need for structural arrangement to accommodate the substitution. Then in the middle, we have a locally unsustainable case. The first neighborhood of the component is too packed to accommodate substitution. However, some structural arrangement allows the accommodation, and this is made possible by the fact that there is empty space at the second shell neighborhood around the component. Finally, in the last case, we see that space is slightly occupied everywhere in the system, and so structural arrangement is impossible to accommodate any substitution. So we say that this last case is unsustainable at the multi-scale level according to the definition. So the fact that space is tightly occupied in the unsustainable case is represented in the complex networks by the presence of high-weight links, and in particular, when chains of adjacent high-weight links are present, it means that space is tightly occupied everywhere in that direction, and this is an indication of unsustainability, so the impossibility for performing the arrangement according to this representation. So we have looked for adjacent high-weight links in the networks as an indication of unsustainable situations. In the urban cases that we saw before, again, we found both sustainable and unsustainable building layout solutions according to the definition. For example, Mont is classified as sustainable, as the high-weight links, the red ones, in the building network are well scattered in the system and rarely adjacent one to another. We interpret this by saying that some empty space is usually present around the building to accommodate for structural arrangement. Instead, in Schabanel and even more in Manhattan, we see chains of adjacent high-weight links that are an indication of unsustainability, according to the definition I gave before. In proteins, again, we have never found chains of adjacent high-weight links of the size comparable to the size of the system, and so we will say that proteins are sustainable also at the multi-scale level. So to summarize this first part, we have found that proteins are classified as sustainable as space is never tightly occupied around amino acids, and this is true at the local but also at the multi-scale level. In urban cases, instead, we have seen that the situation is really case dependent. there exist sustainable building layout solutions at Mont as we saw before, but there are also unsustainable cases. That means that parking is very tight in these regions. And so substitutions of buildings will be hardly accommodated. So I will not go any deeper into the analysis of urban systems. I just want to highlight once again how this measure of space occupancy through the building network allows drawing some conclusions on the impact of different building layouts on the possibility for performing substitutions. I will now go on with analysis of protein structures, for which I had said that amino acid substitutions should be accommodated, and this can happen through structural arrangement or not. So to have a better idea on whether this happens or not in proteins, I have performed in silico substitutions of all the amino acids of a protein. So each amino acid was substituted by all the other possible types of amino acids. I will not have the time to go into the details of this analysis. However, I would like to show this result, that is the table you can see in the bottom of the slide, where each column corresponds to an amino acid position and each row corresponds to an amino acid type. So each cell corresponds to an amino acid substitution. And the cells are colored based on the scale of the arrangement they provoke. This can be node arrangement, the yellowish ones, a local rearrangement, or a large-scale rearrangement. So from the fact that all these colors are present in the table, we can see that the response of protein structures to substitutions is highly complex, and it confirms the fact that substitutions can be accommodated with and without structural arrangement, and this can involve multiple scales. So to conclude for the first question, we have seen that the possibility for accommodated substitutions and the need and the scale of structural arrangement depends on the local to global space occupancy in the system. And the good news is that we can measure the space occupancy thanks to the complex spatial network models. We can now pass to the second question, that was what is the impact of rearrangement on the system dynamics. As I said, I will continue only on the analysis of proteins for this question. And we understand that answering this question is very tricky if we only use data coming from the crystalline structure of proteins. Indeed, this question somehow implies that there is some dynamical information in the static picture provided by the amino acid structure and its amino acid network form. I have proposed a measure in this sense, and to arrive to its definition, I need to provide a couple of more information on protein structure and dynamics. The first point that I want to underline is the fact that protein structures can be made of multiple amino acid chains. In this case, we speak about oligomers. And in here, I represent the same protein structures that we saw at the very beginning of this presentation, but using a different way to represent its atoms, we can recognize the existence of 5 chains. So in this case, we say that the protein is a pentamer. The second point I would like to highlight is that we should not forget that the 3-dimensional structure of a protein is made by the folding of a 1-dimensional amino acid chain. So somehow the amino acid sequence of the protein gives some constraints of what are the possible neighbors in space of an amino acid in the final structure. So for example, here I look at the spatial neighborhood of one amino acid, number 74 in the amino acid sequence, that is highlighted in black in the figure, and in gray I have all the amino acids that are at distance lower or equal than 5 angstroms, so they are neighbors to 74 in the amino acid network. We can classify these neighbors based on the distance from 74 in the amino acid sequence. So we'll have 1D neighbors that are just on the right and on the left of 74 in the sequence. Further, we'll have 2D neighbors. Even further, we'll have 3D neighbors. In this case, this 3D neighbor is next to the 2D neighbors, but this could be elsewhere in the amino acid sequence. And finally, we have 4D neighbors that belong to a different chain. This classification highlights 4 structural levels in a protein, 1D, 2D, 3D, and 4D. And this is important because these different levels are roughly associated to different dynamics in the protein. So the classification I propose here on the right is very rough. However, the important message is that 1D contacts are associated to fast motions that involve only few atoms of the amino acids, while 2D, 3D, and even 4D contacts control dynamics that involve many atoms and so are slow motion. For example, the 4D contacts control the motions of different amino acid chains. So these are movements that involve the whole chain, so a lot of atoms. So it's large spatial scale and slow motion in time. So because of this relation between the structural levels and the atomic dynamics, I have proposed to look at how the atomic interactions are allocated to the different structural levels around a given amino acid. So the total number of atomic interactions made by an amino acid is measured by the node weight. And so I have proposed to look at how this node weight is partitioned in the different levels. So in this case, for example, we have that 31% of the atomic interactions belong to the 1D level, so it's the sum of the link weights belonging to the 1D. 61% belong to the 2D, only 1% to the 3D, and 6% to the 4D. And so the hypothesis is that if these fractions or percentages change because of a perturbation of the system somewhere, then the atomic motions around 74 should be different. To verify this hypothesis, I have compared this allocation measure made by amino acids that belong to protein variants that have similar sequence, same structure, same function, but different dynamics. So I have defined this delta Fix measure, where Fix is the fraction of atomic interactions allocated to the X level by amino acid of position I in the sequence. So I've applied this comparison to 3 case studies, and today I will present the case of TTR variants or transligative. TTR is a protein present in the human body, and it is highly studied because it is associated to some diseases when it passes from a globular shape to an amyloid fiber shape that is elongated. So this is part of the Alzheimer type diseases. In particular, there exist some substitutions of amino acids that make TTR more likely to form these fibers. And so accordingly, they are called pathogenic substitutions. So I have compared the allocation made by amino acids in 4 variants of TTR, each of them after a single substitution, with the allocation made in the wild type TTR, that means before the substitution. So in the plots in panel C, the x-axis is the amino acid sequence, and the vertical bars correspond to changes in allocations in the different structural levels. The first substitution, T119Y, is non-pathogenic, so it behaves the same as the wild type. Just as a matter of notation, when I say T119Y, it means that amino acid in position 119 was of type T in the wild type, and it has been replaced by an amino acid of type Y. So I was saying it is a non-pathogenic substitution, and accordingly we find no changes in allocation, apart from a segment, this 9803, but this is a highly dynamical segment, so this change is probably not related to a change in dynamics of the protein. The second substitution, L55P, instead is pathogenic, and accordingly we find changes in allocation. This happens specifically at this 50-58 segment that is highlighted in blue in the panel B. We have looked more in details to the changes in atomic interactions at this segment compared to the wild type, and we have proposed that this segment creates a novel interaction interface between different chains of TTR. Then, based on the position of this interface in the globular shape of TTR, we have performed a one-dimensional tiling procedure that resulted in the model for the amyloid fiber or TTR that you can see in the bottom of the slide. And this model has a diameter that is consistent with the experimental evidence. The third substitution with RTM is also pathogenic, and we do see changes in allocation. And this happens at a different segment, is 86-19-2, that is in right in panel B. And this suggests a different pathogenicity mechanism compared to L55P. Moreover, the fact that this segment is part of the interaction interface between two chains of TTR is consistent with the biochemical evidence that the chains of TTR dissociate before the fiber is formed for this V30M variant. The last substitution, T119M, is very interesting because it is non-pathogenic, but it also has the ability of correcting the error caused by V30M when both substitutions are present in different chains of TTR. And we do find changes in allocation that shows that error correction mechanisms require some perturbation of the system, otherwise there would be no way that they would compensate some other changes somewhere caused by other substitutions. So to conclude on the second question, we have seen that the dynamical impact of rearrangement depends on exactly what atomic interactions are perturbed, and I have proposed to look at the structural levels to which these interactions belong as an indication of possible dynamical changes. Now we can pass to the third question, that is, what is the collective impact of multiple substitutions? I will not have a short answer for this question. However, I would like to present a tool that I think may be useful to investigate this problem in the future. This tool is called the GCAT network, and it is now a directed network where the links are replaced by arcs that are arrows that go from one node I to a node J. In particular, in the GCAN network, an arc from I to J means that when I is substituted, then J changes neighborhood in its amino acid network. So we say that I has perturbed J. But we'll give you an example. Here, I zoom in the structure of a protein, and I compare the wild type with the structure after the G330T in silico mutation. And we can see that after the substitution, the link between amino acids 372 and 336 is lost. And so we say that 330 has perturbed 372 and 336, and we put the arcs in the GCAN network accordingly. As another example, after the A375Q in silico substitution, the link between 375 and 330 is created. And so 375, that is substituted, as perturbed 330, and so we put the corresponding arc in the GCAT network. And then we repeat the same procedure for all the amino acids and all the possible substitutions in the protein. You can see that from the point of view of node 330, we now have two types of arcs. Arcs that leave the node, they are called out arcs, and arcs that enter the node, they are called in arcs. So accordingly, we can define two node degrees. The in degree is the number of arcs that point to a node. That is the number of substitutions that can perturb that node. And the K out, the out degree, is the number of arcs that leave the node. That is the number of amino acids that are perturbed when the node is substituted. According to these measures, I have classified the nodes in the GCAN network into four classes. G generate when K out is large and K in is low. The contrary is absorb. The K in is large and K out is low. T transmit when both node degrees are high, and C connect when both degrees are low, and high and low are defined based on the average K in and K out in the network. So you can see here the whole GCAN network of a protein, where the nodes are colored based on their classes, and you can see that all classes are populated. This indicates that the quantity of perturbations caused by substitutions is very amino acid position dependent. Moreover, we have not found any easy relation between the class in the GCAN network and whether the amino acid is on the surface or buried in the protein structure, and this suggests a complex design for the structural response of proteins to perturbations. So something important about the GCAT network is that the relations modeled by its arcs go beyond the spatial relations that are modeled by the amino acid network. For example, here I highlight the outer neighborhood of an amino acid 308 in the GCAT network. These are the amino acids that are perturbed when 308 is substituted. Among these amino acids, some are first shell neighbors of 308 in the amino acid network, it's the green nodes in the figure, but others are second shell and third shell neighbors, the orange and red ones. That means that are further from 308 in the protein structure, as you can see in the representation on the top right. So this once again shows that the rearrangement caused by amino acid substitutions can involve the large scale in the protein structure. So why is this interesting for assessing the impact of multiple substitutions? The GCAT network is created starting from the information on single amino acid substitutions. However, it results in a global network that connects all amino acids of a protein. So I propose that we could use the GCAT network to look at what amino acid substitutions should act collectively in the system compared to the ones that should act independently. And I will give you an example using the case of error correction mechanisms that, as we've seen with the case of TTR, these are possible means to, as we have seen with TTR, error correction is possible with multiple substitutions. So let's say, for example, that we want to correct the perturbation caused in amino acid 395, the red one, by the substitution of amino acid 308, the black one. We could use the GCAT network to look at the amino acids that also point to 395, because these amino acids, when substituted, have the potential, they act on the same region as 308, and so they have potential to compensate the first perturbation. However, we could also look at the amino acids that point to 308 as they perturb the source of the perturbation that we want to correct, and so they could also be investigated as potential error correction substitutions. And finally, we could imagine more indirect relations, as for example, some node that is connected to a node that is connected to 395. This is called a path in the GCAT network. And using this kind of tool, we could now draw hypotheses and test these substitutions experimentally to check whether they could provide some error correction mechanism or not. So to conclude, I have shown that network tools are useful to investigate spatial sustainability, and they have the advantage of providing a high level of abstraction that allows comparing systems as different as proteins and cities. Moreover, they naturally take into account the multi-scale aspect of spatial occupancy, and this is relevant for assessing sustainability, as we saw in the previous examples. For what concerns protein sustainability, we have seen the role of link weights in determining the structural and dynamical robustness of the system, and I have proposed ad hoc measures to investigate this issue. As a perspective, we have seen that we now have to understand how multiple substitutions act collectively to provide, for example, error correction mechanisms. This is relevant for designing drugs that could that could act against pathogenic substitutions, but this is also relevant to design adaptable systems where similar error correction mechanisms could be implemented. Moreover, there will be soon the need to pass to the functional diagnostic of substitutions in order to be able to discriminate alternative solutions from the ones that provide functional innovation. And this is relevant, of course, with respect to pathogenic substitutions, but also for the evolvability of the systems. For what concerns urban sustainability, we have seen that the multi-scale space occupancy is relevant to determine the structural robustness of the system, and thanks to the amino acid, the building network, we can measure the space occupancy and draw some conclusions. As a perspective, we may now move to the dynamical robustness that may be defined as the possibility for maintaining flow of cars or pedestrians in the cities despite substitutions, but we could also conceive some algorithm that may allow generating automatically sustainable solutions, because we have seen that this is possible, and this may be performed under some constraints as buildings, shapes, or distances. Finally, we may add some other parameters to the building network as the building height to study other aspects of sustainability. For example, the possibility for flow of air that is related to the risk for accumulating pollution, but this also may indicate the quantity of solar radiation that can be absorbed by the building, so the possibility for using solar energy, but also the quantity of energy that is expected to be consumed in the building. So I have not used this parameter in the definition of the building network right now, because I think it was not necessary for assessing structural robustness the way I have studied it in here. So I just finished with a list of publications associated to this work and of talks and presentations that I have performed, and I thank you very much for your attention. I'm ready to answer your questions.
Thank you very much.
Thank you.
Okay.